Welcome, everyone. It's lovely to see all of you here. Maybe we can start with a bit of an intro. I'll start. I'm Arushi. I'm one of the co-chairs of the competition and co-moderating this session, and I'm a master's student, and I'm joining from Canada. Sapo, would you like to go ahead?
Yes, I'm Sapo. I'm currently a PhD student at Oxford. I am also one of the co-chairs, and I'm so excited to be here.
Katie, would you like to introduce yourself?
Yeah, of course. Yeah, I'm Katie Schretter, and I'm a postdoc in Jerry Rubin's lab, and will be actually starting my own lab at SMU in Dallas, Texas, but currently joining from Virginia in the United States.
Cool. Anyone in the audience would like to introduce? I can start maybe. I'm joining from UK.
While Sapo's added, maybe you can. We are curious to know actually what makes you interested about today's talk.
Yeah.
Oh, we have someone. Mariana is joining from Canada as well. Nice to see you.
Welcome.
We have someone from Kyrgyzstan. Ubaid, nice to meet you.
Welcome.
Salman, I'm joining from IUK ISM, located in Kyrgyzstan.
Welcome.
Hi, Nayela from Kyrgyzstan.
Awesome. Welcome.
Very global audience today.
Yeah,
Yeah.
it's great.
Yeah, very exciting.
From South Africa, Cape Town. That's excellent.
Awesome. Welcome.
Okay, maybe I'll say why I'm really excited about today's talk. It's admittedly a bit now, the most scientific reason, I really think it's so cute the way flies fight each other. So I'm really excited about the videos. I'll be honest about that.
There will be a lot of videos, so that will be, and and you guys can feel free to talk or if anyone has questions during the talk, please let me know.
Yeah,
But yeah, the videos are always fun to look at as well.
absolutely. I'm curious, this is your own video, right?
Yeah, so this was actually taken on my benchtop
Hmm.
at Janelia. So Igor Schwanovitz took this with Wyatt, and we took it on a very, very high speed camera. So it was about, I think that this one, the camera can go up to 2000 frames per second, but this was recorded at 250 frames per second. So yeah, and we zoomed in obviously very heavily to be able to see them interacting, see them fighting.
Yeah.
Because sometimes I forget the fly is actually really small and the brain
Yeah,
is even smaller. So like it's fascinating how you can like study the tiny neurons and uh,
yeah. I know
Yeah.
it's very, very fun. Yeah. Yeah, and they do these complex behaviors. I mean, they do, which I'll show more about.
Very exciting.
Yeah.
Ubaid says this is really cool. 100%.
Awesome.
Very
Thank
cool. How these tiny things fight.
you.
excellent. Arushi, you want to get us started?
Sure, I'll get us started. So I'll do a bit of an intro and then Sapo will talk a bit about other stuff and then Katie can, lead the stage. So
Awesome.
just to start off, hi everyone. It's so lovely to see everyone here today and so cool to see people from so many different places. That's so nice to hear. But anyways, I'm Arushi. I'm an MSc student at the University of Toronto in the Grintman and the Anwriter labs. I heavily leverage the Drosophila connectome in my own research, and I'm joined here with Sapalnach, who is our co-chair and PhD in Rhodes Scholar from the University of Oxford, and both of us will be co-moderating the session today. We're really excited to have Dr. Schretter give a talk for the 2025 FlyComp2025 Guest Speaker Series, and a bit of an introduction about them. Dr. Katie Schretter was a research scientist at HHMI Janelia Research Campus, and now it's really exciting. She is an assistant professor at SMU, so congratulations. Dr. Schrader is interested in understanding how the nervous system, in combination with genetic and environmental factors, regulates social interactions. Her work has identified neuronal circuitry modulating female aggression, and with collaborators uncovered cell types regulating persistent female aggression and neural circuits linking social states like aggression to vision. Firstly, I would like to thank so much to our audience and the diverse learners that we've had for joining us today. We're really excited. And of course, we're really grateful to Dr. Schrader to be here and sharing their wonderful work. Now I'll pass it on to Sapo, who will debrief us on how the session will be running.
Excellent. Thank you so much, Arishi, for that lovely introduction. Of course, thank you, Katie, for being here. But also thank you to everyone here today, whether you're joining us live or catching the recording later, which will be uploaded on YouTube. And it's so exciting to see people tuning in from all around the world. And I see South Africa, Kyrgyzstan, Kazakhstan, Pakistan, and many other countries. So for those who may be new, this event is part of the 2025 Fly Connectome Research Competition, or FlyComp 2025. So the idea behind this initiative is that we want to introduce young students to the world of neuroscience and synapse-level connectomics. And the idea is that maybe students will get a chance to gain hands-on research experience with using cutting-edge tools such as NeuroGlancer, which is for visualization, and Newprint Explorer, which is for analyzing neuronal connectivity. And through this speaker series, including Dr. Schretter, we aim to deepen students' engagement with connectomics, allowing them not only to hear cutting-edge science directly from leading researchers, but also to understand how their own research journey with the Fly Connectome connects to the broader questions in neuroscience. And I think Dr. Schretter will be talking about the optic lobe connectome as well, which is the data set we'll be using for the competition. So that will be exciting. It's exciting for me, and I'm sure Arushi as well. And I would like to also thank our partner organizations and ambassadors from all around the world, including Libya, Pakistan, Kyrgyzstan, Kazakhstan, Egypt, Nigeria, South Africa, Canada, and the United States, who have all joined our mission in making neuroscience more accessible to everyone. And of course, once again, huge thank you to the audience who have shown up today and taken the time to learn about neuroscience. So, before we dive in, perhaps you can rename yourself on Zoom, and perhaps you can also add a tag after your name that best describes you. So if you're a high school student, undergrad, postgrad, postdoc, faculty, research enthusiast, this might help us better appreciate the diversity of learners in our audience. Or feel free to type in the Zoom like where you're from, which I know you have already been doing. In terms of structure for today, I think Dr. Schretter will begin her talk, and afterward we'll move into Q&A session. You're welcome to drop your questions in the chat as they come to you, or you can also raise your hand, and Arusha and I will try to keep track of them throughout and moderate. And once we get to the Q&A, we'll also open the floor so you can raise your hand and ask your questions directly. And again, like I said, we love informal discussions, so I don't want you to get scared or anything. And with that, we're thrilled to get started. So, Dr. Katie Schretter, the stage is yours, and we are all excited to talk.
Awesome. Well, thank you guys so much for that introduction, and thank you again to the audience for joining from all over the globe. It's amazing to see so many people and to hear that so many people from across the globe are joining in this effort. As we were talking about, it's amazing to see the community all come together to do this. So I'm really excited to present my work to you today. And so today I'll be talking to you guys about my work in the Fly, really looking at social behavior. And so the way in which I like to kind of look at this is social behavior in the context of aggression in the fruit fly Drosophila melanogaster. And so just to kind of orient you to social interactions, when you might think of, social interactions commonly like these ones, you're at a festival or at a farmer's market or fair, or, out with your family or friends. And so you're interacting with other individuals. And so that is, typically how we think of these social interactions. And they're ones that are not only just important for us, but are important across many different species and across many different, attributes of our daily lives. And so, just to kind of think more in depth about these questions, I'm going to focus in on these two individuals here, which we can see, they're probably eating, they also are talking with one another. Those aspects of the environment are things that are really critical for these social interactions. So, as neuroscientists, things that we think a lot about is, one particular question that we think a lot about is, how does the environment really shape these social behaviors, as well as, the underlying neurons and neuronal circuits in the brain and that contribute to regulating these behaviors. And so in terms of the environment and, how it regulates social behavior, there's many different attributes of the environment that can have an effect. One of them are, all the sights and sounds and smells that might be going on, which, are things that we like to refer to as sensory cues. So you guys have been looking a lot at the visual system, which, kind of sights, but then there's also other information that can also come in as well. And so all of this information is coming in at one time, at different times, and also can be processed within the brain brain. And so we like to refer to that as multisensory processing. And that is something that is actually known to be related to many different disorders. So things like autism, as well as other, neuropsychiatric disorders. And so understanding how all of these different senses are processed within the brain is a subject that's of importance, for basic science as well as for translational sciences as well. And so in addition to all of these sensory cues, another important aspect of the environment is that of the internal environment. So what do I mean by that? Well, I mentioned that these individuals were eating, and hunger is one thing that we like to refer to as an internal state. And so there are other internal states, things like aggression can sometimes be referred to as internal states. And how exactly they're processed and also structured in the brain is a subject of intense focus for a neuroscientist. So there's been many different theories that have been proposed, one of which has been hierarchical structuring of these internal states. And, as neuroscientists, we really want to understand, are those theories correct? So, how exactly is this actually being implemented in the brain? And so those are all central questions in neuroscience that we've been really focused on looking at and have been looked at across many different species. And so the one in particular that I think is very amenable to looking at this on a cell type specific and brain-wide level is that of the fruit fly. So just to kind of think more about this and about why, I in particular like the fruit fly for this. One of this is really genetics, which I'll get a little bit more into. We can go in on a really cell type specific level as well as molecule specific level to manipulate individual cells and molecules. We also can utilize, like you guys have been, these maps of the brain. So we have a full map not only of the female, but actually now we have, that has been recently released, we have the male as well. And so we can look across sexes and really look and see at similarities as well as at differences. So really look at the variability that's there. And then we also have a lot of methods for looking at behavior and physiology, so looking at the activity of the neurons. And so those are all things that we can do in the fly and that I'll be talking about. And so this question of how does the environment shape social behavior is one that's particularly motivating for me in terms of, my science, as well as also, as one that can be applicable to basic science and to therapeutics as well. And so in terms of social behavior, one critical one is that of aggression. So aggression is exhibited across many different species. It's also exhibited across sexes. You can see it here with these female fruit flies. My sister actually recorded this video in the wild in the United States. And so when you see these types of behaviors, the individual actually engages in them in order to gain a certain benefit. So gaining access to a territory or a mate, or potentially, a certain reproductive area. And so those types of behaviors, while they carry with them those benefits, they also carry with them considerable risk. So risk in terms of risk of injury, as well as also potentially death. And so, therefore, they need to be really tightly regulated in the brain. And so some of this regulation can come from those external sensory cues, as well as also internal states. And so those are some of the ways in which aggression is regulated. Additionally, also, there is different phases to aggression, and this actually can be found across different species, but I'm going to showcase it in flies. So you can see that there's an approach phase, so where one individual will come towards that of another individual. And then following from this, there's usually an engagement phase, or I like to think of this as the fight phase, where two individuals are engaging in those aggressive interactions. So here I'm showing you one particular behavior that's primarily exhibited in females. So it's a head-butting interaction where one individual uses their head to push that of the other individual. Another behavior that's an engagement behavior is that of this lunging behavior where the males will strike down on the other male. And so following from these, engagement activities, there's usually a time in which an individual will move away from one another. So I like to think of this as a disengagement or a retreat phase where one individual moves away from the other individual. And so although these different phases have been studied across many different species, and there's been many different labs that have really focused in on this, there's still some outstanding questions that remain in the field. So one major outstanding question is, what are the neurons that modulate the attack, this fight phase? And I mentioned the fact that, in fruit flies, there's sometimes differences across sexes in terms of how exactly they engage. And so, what another critical question is, how are these similarities and differences, how are they mirrored in the brain? And so what are the differences and similarities across sexes? In addition to this, I talk a lot about the kind of the escalation of aggression, but really, another critical question that I think is of heightened importance is, what are the neurons that really modulate the disengagement or retreat, so really downregulate aggression? And so these are really critical questions that, at the beginning of my postdoc, I really sought to answer. And so I'll be walking you throughout the talk through these different questions. And the way in which I sought to, or what we really needed to address these is we really needed an organism that had complex aggressive behavior, had the ability for us to analyze them, had some capability to go in on a cell type specific level and manipulate, neurons in the brain, and also had the capability, we thought it would be really nice to have a full map, because then we could have a full picture of how these neurons all connect to one another in the brain. In the fruit fly, we have a lot of these tools thanks to a lot of work that has been done across many different labs. So in computer vision and machine learning, there's been some beautiful work that has been done to be able to develop algorithms for tracking and analyzing behavior and doing this automatically, so using the computer to do this. Also, beautiful work done in genetics to be able to develop really specific tools that we can use. And then now we have this wiring diagram that you guys have been using. So we now have the capability to not only develop these hypotheses behind how exactly these behaviors are being regulated, but we can actually constrain the hypothesis space. So we can constrain it down to specific neurons and specific cell types to really then go in and test those cell types. And so this is all work that I've been doing during my postdoc. And so I'm going to walk you through, at the beginning, I'm going to walk you through kind of the behavioral work and the genetic tools I've been using. And I'll come back to a little bit in the mid of it, the Connectome. So just to start you off and just to orient you to what I'm using for my behavioral analysis, I'm using a setup like this one. Okay, so what I'm using is a behavioral setup like this one, where we have a camera that basically is overhead, so I can record the flies from overhead, and then I can use lights, so basically the flies can visualize, they can see one another, and I can turn off the lights so we can test, if in the dark they have similar behaviors and things like that. In addition to this, I can also utilize LEDs underneath. And what those LEDs can do is I can shine light and different wavelengths of light to turn on and off particular neurons through using these things called light-gated channels. And that is called optogenetics. And then in addition to optogenetics, I can also use another thing called thermogenetics, where basically I use temperature to essentially then turn on and off neurons. So I can use these temperature-gated channels to essentially then, when I basically increase the temperature, I then activate these particular neurons that I have. So I can use both optogenetics and thermogenetics in this setup. And so when I started in the Rubin Lab, I was primarily looking at these neurons, which are called aIPg neurons. They're just so named because of their cell lineage. And these neurons are roughly around about 11 neurons. They are all cholinergic, so they're all excitatory. And they actually express a particular gene called fruitless, which is a transcription factor that actually causes the neurons to be different in morphology across sexes. And so they differ across sexes. Here I'm showing you the female version. And these are only 3 of those that are found in females. Or 3 of the types that are found in females. And so these particular neurons were of interest to us because actually before I started at Janelia, we had found a really interesting phenotype. So to quickly orient you to the fly's behavior before activation, what the flies are doing is you can see them reaching out and touching one another. You can see them jump. And this is typically what they do when they're in that behavioral assay or that behavioral rig that I showed you before. And so this is prior to me activating those neurons, those aIPg neurons. Now, what I'm going to go in and do is I'm going to activate those neurons through using a thing called a light-gated cation channel. So basically when I shine red light, then the channel will open, causing the neurons to fire. And so when I do this, what we see is that now the flies start to engage in these really tight, aggressive interactions. And so you can see that very clearly here. Now, if I zoom in on that, you can see that the flies are engaging in these things called the head-butting behavior that I mentioned, so that you can see them using their heads to basically push one another. You can also see them fencing with one another, which is where they're reaching out and touching each other. And each of those are aggressive interactions. And what had typically been done in the field is that individuals had gone through and manually annotated each individual frame as whether the flies were engaging in aggression or not engaging in aggression. And since I wanted to screen through a ton of videos, so I wanted to look at many, many different videos, what I needed an automatic way of doing this. So I collaborated with Kristen Branson's lab here at Janelia. And what we did was we set up a behavioral analysis pipeline that they developed, and I have now co-opted to be used for this. So what we do is we can track the flies over the course of the entirety of one particular setup. And so basically here I'm showing you two trajectories. And so this one is one in yellow, and here's one in red. And I can track actually all the flies in this arena. And then what I can do is, using a thing called a classifier, I can have the computer go in and automatically score whether or not the flies are engaging in aggressive behaviors or not engaging in aggressive behaviors in each individual frame. So I can go through many, many different videos through doing this. And so when we do this, what we find is that when I activate those neurons, you can see that I get an increase in female-female aggressive behavior. And so you can see that here. Now, what you might also notice is that not only do I see an increase in female-female aggressive behavior during the activation period, I actually see it after that period. So you see it continue. Now, this is really interesting in light of these things that I mentioned called internal states. Well, just to back up a step, what is an internal state? Well, an internal state is the thing called a partially hidden variable. Now, one critical feature of them is that they continue or extend beyond the initial stimulus period. And so a good example of this is hunger. Since it's around about 9:30 my time, I usually eat breakfast. And so if I see an apple at this time, I'm going to start to get very, very hungry. And that hunger is going to continue beyond just, me, seeing that apple. It's going to continue even if I turn away or go into another room. And so it will continue beyond the stimulus period. And so we've seen that when we activated those aIPg neurons. And so what, was a critical question is, how is this occurring? Well, the cell types that were known to be involved in terms of aggressive states in flies were primarily known in males. So we knew about these neurons called P1 neurons, which I'll talk a little bit more about later in the talk, but these P1 neurons actually promote a male aggressive internal state. Now, a lot less was known about that in females. And so we thought, okay, maybe these aIPg neurons that we've been studying, maybe they not only just promote aggression, maybe they promote an aggressive internal state. And so in order to really look into this further, what I did was I collaborated with Vivian Chu, who at the time was a graduate student at Caltech in David Anderson's lab. And we did a series of experiments that I'm going to just really quickly walk you through. So in these experiments, what we did was we had two flies basically in an arena, which was separated by this sliding door here. And so this sliding door is one that is basically the flies cannot see one another. And then what we can do is we can activate those aIPg neurons through using those red light. And so we activate them while the flies cannot engage, they cannot see one another. And then what I'm going to do is what we did is we left the flies in this particular setup with the sliding door for 10 minutes and then released the flies to engage with one another. Now, what we're asking here is, if aIPg is in fact promoting an aggressive internal state, then what should happen is that the flies should basically hold on to that aggressive internal state over the entirety of those 10 minutes, such that when I release the flies to actually engage with one another, they'll start to engage in a high level of aggressive behavior. And so that is what should happen if aIPg is in fact involved in promoting an aggressive internal state. And so what we found was that actually, in fact, aIPg is doing this. So what I'm showing you here is this is called an ethogram. What it is basically showing you each an individual instance of aggressive behavior. So that is shown in this red ticks right here. And basically what we did was we stimulated those aIPg neurons, those particular aggression neurons, and then left the flies isolated for 10 minutes. So the flies cannot see one another, they cannot engage engage. And then when we removed this door here, we now see that the flies engage heavily in aggressive behaviors. And we see that above that of the control. And so this really showcases the fact that aIPg, these aggression neurons, are not only just promoting female-female aggressive behaviors, they're actually promoting a persistent aggressive internal state. And so, we think that they're promoting this aggressive internal state, but the next question is, okay, well, how exactly are they shaping behavior then? When we think about this, I mentioned the fact that these aggressive states are regulated by these external sensory cues and these internal states. Now, when I showed you these two flies about to engage in aggressive behavior, I showed you just two of the flies. This is not typically what you find in the wild. Usually it's more, akin to, when you're out at, a particular farmer's market or at a festival. I think we have a question. So this is typically what you would see, is that there are many different species. There's many different other individuals, and so the flies really have to be able to focus in on one another. So they have to really be able to attend to one another. So how do they exactly know what to attend to? This type of a question is actually one that is more general question. When you are driving or walking, you have to really be able to focus in on what's in front of you and tune out the rest of the surround. So you really have to have that focus, that attention when you're actually engaging in these types of interactions. And it's not just aggressive interactions that require this attention, it's many, many other interactions. And so this is more of a general question in neuroscience, is how exactly do they know what to attend to? And for flies, vision, as you guys have been looking into, is one that is particularly critical. And so that was one that we were really interested in. But, one question that we actually didn't know was we didn't know whether, in fact, vision is actually involved in female aggression at all. And so we needed to back up a step and really ask the question, is vision important for these female aggressive internal states? And so, in order to ask this question, what I did was I placed the flies on a mutant background. This is a phototransduction mutant. So what I'm doing is I'm rendering the flies fictively blind. And then what I'm going to do is I'm basically going to activate those aIPg neurons. Now, if vision is important for these aggressive behaviors that are mediated by aIPg, then I shouldn't actually see that much aggressive behavior when I remove all these visual cues. And so when I activate these aIPg neurons under these conditions, you'll see that the flies will reach out and touch one another, but they won't continue to engage in those aggressive behaviors. And so this really showcased that vision really seemed to be important for these continued aggressive behaviors. And so the next key question was, okay, well, I removed all the visual cues here, but what visual cues might be important for aggressive behaviors or important during these aggressive behaviors? And so, what are the visual cues that the flies are experiencing during these interactions? And so, one critical feature is that of the angular size of the other fly. Now, what this just means is it just means the amount of space in the field of view that the other fly takes up. And so what I did was I wanted to see, what are the angular sizes that the flies are experiencing during these aggressive encounters, because that'll give me a little bit more information to be able to understand exactly, what particular neurons might be involved. And so what I found was that the flies were typically experiencing other flies basically very, very close to them. So it was primarily a moving object that was roughly around about the size of 15 to 30 degrees in size. So these are objects that are super, super close to the other fly. And so, this was really interesting to us because of the fact that we, based on all of the literature and all the beautiful work that has been done by many different labs in the fly community, we now have a little bit more information about how flies process these visual cues. And so, there are these things called visual processing neurons, which some of you might be looking into. And so these are ones like these lobular columnar neurons, which are tuned to specific visual features. And so some of them are tuned to these looming objects. Others are tuned to things like fly-sized moving objects. And one in particular that was of interest to us is that of this LC10A neuron. So this LC10A neuron has already been shown to be involved in terms of male courtship, and specifically when a male is tracking a female during courtship. And so that was particularly intriguing to us because it suggested to us, when a male is tracking a female, it's probably, roughly around about the same size or maybe about the same size as a female is experiencing during these aggressive encounters. But we wanted to actually measure that. So we wanted to see whether the same sizes that are important for these LC10A neurons, whether there was any overlap with that of what the flies are experiencing during aggression. And so through a series of experiments that was done by Nathan Klepke in Michael Reiser's lab here at Janelia, what we found was that actually the preferred object sizes that are found for these LC10A neurons actually do have overlap with that of what the flies are experiencing during aggression. And so this was quite striking to us because it suggested, okay, maybe these LC10A neurons might be involved in contributing to these female aggressive behaviors. And so that was our next critical question is are these LC10A neurons, are they important for aIPg-mediated aggression? Now, in order to talk more about these experiments, I'm going to back up a step and just walk you through how exactly I do them. So, when we do these experiments looking at aggression, what I'm showing you here is I'm showing you the average aggression that the flies are exhibiting during a 30-second period. And this is in this particular assay. And so the baseline level of aggression is rather low. It's roughly around about 5%. And so if we're looking to see if there's going to be a decrease in aggression, it might be really, really hard to see this because we're already at a particularly low level in this certain assay. So we might not be able to see any decreases. So therefore, what I needed to do is I needed to boost up this baseline level of aggression. And so the way in which I went about doing this is I actually constitutively activated these aIPg neurons through using a temperature sensitive channel. So basically when I boost up the temperature, that opens the channel and activates the neuron. So we now at this level, you can see that we have a high level of aggression when I activate these aIPg neurons. And that basically allows us to now not have these basement effects where basically we can't see any change in aggression. And so what I next did was went back to this question, okay, is LC10A important for aIPg-mediated aggression? Now, we have this boosted level of aggression when I activate aIPg, but what this circuit model predicts is that, if I inactivate LC10A, I should get a decrease in aggressive behavior because basically, if I'm still activating aIPg, I'm getting this boost, but if I'm inactivating it here, I should see a decrease in aggression. And that is in fact what I do find. And so it does suggest that LC10A is actually mediating these effects via aIPg. And so this was very nice for us because now it suggests that we have, a specific pathway that we can look at in terms of these visual neurons. And so we wanted to ask, okay, how is LC10A connected to these female aggression neurons, to these aIPg neurons? Now, in order to do this, I needed to actually turn to the connectome. So I'm going to walk you through, how exactly we did this for my project. So a lot of this, actually, I should mention, some of this started way before I even got started at Janelia. So they were doing some quite beautiful work as a team to basically trace through the entirety of one hemisphere of the female fly brain. And this took a lot of concerted effort from many, many different individuals at Janelia, as well as at other institutes. And so what we were doing is we were taking these light microscopy images of these aIPg neurons and then trying to match them to these electron microscopy data sets. The way in which we do this is, some of you might have seen this in Neuroglancer, is that we take, we have these little slices of the fly brain. We can trace these neurons throughout these axon bundles throughout the fly brain. And then we can generate these particular very, very beautiful skeletons of these neurons. And we can basically match them between the light microscopy images in order to see whether or not they are an exact match. And so this was not only being done for my neurons of interest, but we're actually being done throughout the entirety of one hemisphere of the female brain. And so we could look at not only just these connections between these aIPg, between these female aggression neurons, but we could actually identify all of the inputs to these neurons, as well as all of the outputs of these neurons. And so the reason this is quite striking is because, like you guys have been seeing, is with the iConnectome, we could basically now trace all the way from sensory information coming in all the way to motor output, which is something that when I was a high school student or even as a college student, I did not think that this was possible. And so it's something that, I like to think of as kind of a golden age for neuroscience, particularly in the fruit fly, because now we can really generate these quite beautiful hypotheses and we can do amazing amazing work across many different labs in order to actually, in many different places, in order to actually ask these key questions and get into a little bit more about, how exactly the brain is working in order to create these really complex behaviors. And so that can all be done as a team and as a group and as a community. And so going back to the central question, okay, how is LC10A connected to this female aggression circuit? Well, when we were doing this and searching through, what we actually found was that these aIPg, these aggressive state neurons, were actually connected to these LC10 neurons in 3 separate ways. So it really looked like these aggressive state neurons were actually modulating vision in 3 different mechanisms. And these mechanisms actually are some of the top downstream outputs of aIPg. So it really looks like they're really heavily focused on modulating vision. And so I'm going to really quickly walk you through, each of these different mechanisms, a couple of experiments about how exactly we tested them. Because while we had, these circuits, we wanted to actually test their functionality and test their function in aggression. And so, the first mechanism we like to refer to as a toggle switch, basically, kind of like a light switch, basically essentially amplifying these particular LC10A neurons, which are the neurons I was mentioning that are involved in terms of moving object detection, and then serving to dampen these LC10C neurons, which, are predicted to be involved in terms of color vision. So just to walk you through this particular circuit model, what we're saying is that basically what the circuit model predicts is that basically when aIPg, when these aggressive state neurons are activated, what is serving to happen is, aIPg is excitatory. These 2 neurons I'm showing you here are actually inhibitory. And so what it's serving to do is that when aIPg is active, you should serve to basically boost up the activity of LC10A. Now, conversely, at the same time, basically when aIPg is active, through this one particular inhibitory neuron, it should serve to dampen down LC10C activity. And so basically it's boosting up this moving object detector activity while dampening down this LC10C activity, which is one that we predict to be involved in terms of color vision. And so, we had the circuit model, but we wanted to test, whether or not it was in fact functional. And so what we did was, and this is through a collaboration with Mei Xiao, who was in the lab at the time, is we did a series of electrophysiology experiments, basically where we're recording the activity of the neurons. So we're basically sticking an electrode into the neurons in an active brain. And so what we found was that when we do this, when we activate these aIPg neurons and then record from these particular 2 neurons that I was mentioning here, is that we found that it actually directly recapitulates exactly what we would expect based on the circuit model. And so we really think that this is a functional model, but we next wanted to test, is it functioning in female aggression? And so in order to do this, I just did some optogenetic flip of the switch. And when I did these optogenetic activation and inactivation experiments, I found that these particular neurons were actually implicated in female aggression. And so this really showcases that, not only is this particular circuit involved in terms of female aggression, but it actually looks like it's functional as well. And so this toggle switch is the first mechanism that we identified. The second mechanism that we identified is one that we like to call a disinhibition. And this is one because we basically have these 2 inhibitory neurons, and that's why we call it a disinhibition. Now, the one particular neuron that we were looking at was this IB112 neuron. Actually, in your guys's particular, in the male connectome, it's called LOFC1. And so this IB112 neuron is very interesting because actually it is a visual centrifugal neuron. And what that means is it basically reaches back into the primary visual region in the optic lobe to basically then serve as modulator of that primary visual region. And so this is why it's so intriguing, is because actually it's only one of about 70 of the neurons that do this. And it sends roughly around about 90% of its outputs back into this primary visual region. So it really looks like it's heavily modulating it. And so when aIPg is active, really what this circuit model predicts is that it's serving to, through IB112, basically then boost up this the activity of these LC10 neurons. And so what we next wanted to ask is okay, well, is aIPg actually shaping behavior via IB112? Now, in order to ask this question, what we needed a line, a genetic line that was specific for IB112, because we really needed to look at that particular neuron. And so just backing up a step about how exactly we do this in the fruit fly. So the way in which we go about doing this is we take these two particular lines that look really messy. So they actually are marking many, many different neurons in the fruit fly brain. And what we can do is you can see that they have a little bit of overlap in this one particular neuron. So what we can do is we can actually intersect those two. And when we intersect those two lines, you actually get this very, very quite beautiful and striking line that now only labels one particular cell type, this particular cell type of interest. And so it takes a lot of crosses in order to get a line that actually labels one particular cell type of interest. And during many of the shutdowns in the United States for COVID-19, I was doing a lot of these crosses in order to generate lines that were specific for these neurons within the female aggression circuit. And we got really lucky. So we actually got a line or a couple lines that looked like this. So they looked in morphology very, very similar to these IB112 neurons. Now, what the connectome gives us is it not only just gives us the connectivity of all of the neurons and how they connect to one another, but it actually gives us the morphologies of all of these neurons within that hemisphere of the fly brain. And so the reason that this is striking is because of the fact that the fly brain, the morphology of the neurons is stereotyped. So it appears to be very similar across individuals. What we can do is we can actually use those morphologies to ask, is this neuron actually labeling IB112, or is it just labeling another neuron that looks really similar? And so we can basically do a match to see what particular neuron this actually bears very close similar homology to. And so when we do this matching, what we find is that actually IB112 is the closest match. So it really looks like this particular genetic fly line is actually, in fact, labeling this IB112 neuron. And so then we can go back and we can utilize this to really ask this question, okay, does aIPg shape behavior via these IB112 neurons? And so again, I boosted up the baseline level of aggressive behavior. And what I wanted to go in and do is I wanted to basically inactivate IB112, because what this circuit model predicts is that if I inactivate IB112, I should see a decrease in aggressive behavior. And this is, in fact, what I do find. So if I inactivate these IB112 neurons, we actually do get a decrease in aggression. And so this really suggests that not only was that toggle switch functioning in aggression, but actually also so was this disinhibition mechanism. And then thirdly, and finally, the final mechanism that we identified was this one that we like to call shared outputs. So, this particular one is one in which where we have these aIPg, these aggressive state neurons, and LC10 neurons, so these visual object detectors. Basically, some of them go to the exact same outputs. And so it's not just one neuron that they go to, they actually go to a multitude. It's roughly around about 23 different neurons that they both go to. And really, these neurons actually connect to other neurons downstream, descending neurons, that actually are known to be involved in terms of discrete behaviors, things like forward walking and turning. And so what we think that these particular neurons are doing, these shared output neurons are doing, is they might be computing, am I aggressive with, do I see a fly-size moving object? And then transforming that into a motor output. So really, this is a quite beautiful mechanism for really taking in internal state information with sensory information to transform that into motor output. And so these are things that we're interested in kind of looking into in the future of really how exactly this happens and how this happens over different timescales and, what different features of aggressive behavior these particular output neurons are doing. And so, we identified each of these different mechanisms. And what I should say is that, while I talked about them as being, separate, actually there is a lot of overlap between all of these neurons. So it looks more similar to this simplified circuit model where they're kind of, all overlapping on one another. And so that's something that we're interested in, is how exactly do they influence each other? And that's something that we're looking into in the future. And so, what I believe that we've shown is that, a mechanism for how these aggressive internal states, these aIPg neurons, how they actually are shaping behavior, because it really appears like they're shaping behavior via tuning visual pathways. And so, the next critical question that we had, and you might have noticed this, is that when I mentioned the LC10A neurons, I talked about them in courtship behavior. And so this brings up a really critical question about, how is this similar or different across sexes? And, in talking about this entirety of this circuit that I just mentioned, actually these aggressive state neurons, these aIPg neurons, are the primary neurons that differ across sexes. So the rest of the neurons actually appear to be conserved across sexes. So they appear to be isomorphic. And so that brings up a critical question about, how exactly could this be implemented in the male? We know that there are male aIPg neurons, but we didn't actually know anything about them other than the fact that they exist and they were different in morphologies. And so we collaborated with the group at Janelia as well as the group at Cambridge, who was really working on really developing the male connectome. And so this is some very, very new data where what we've done is we basically traced these aIPg neurons in males as well. And so what you can see is that, some of these neurons, they differ in morphology. You can see that clearly here with these male aIPg neurons here. And so they differ in morphology, but they're still present in the male. And another quite striking feature about these neurons is that actually, in terms of their outputs, they appear to share outputs across sexes, which is a quite striking feature and was quite striking to me because it suggested, okay, maybe they might be having some similar functionality across sexes as well. So even though they're really different in their morphologies, maybe they might be having similar output. And so what we wanted to do is we wanted to actually test these male aIPg neurons. Now, what I didn't mention was actually all of the lines that we had before were only expressing in females. And so we needed a line that expressed in males. And so what we got was we actually got very lucky and we found a line that actually labeled aIPg neurons in males. And it actually labeled that type 1 neuron, which is one of the types that I had found to be involved in terms of female aggression. It also labeled two types that are actually only found in males. So this is male type 2 and male type 4. And so what we could do is we could actually activate these neurons to see, what types of behaviors they might be implicated in. Do we see any aggressive behaviors if we activate these neurons? And so when we activate these neurons, which is when the red light comes on, you see that the flies now start to engage in aggressive behaviors. And this is at 2 times speed. So you can see the flies engaging in different aggressive behaviors. You can also see them chasing one another as well. So it really suggested to us that maybe aIPg in the male is also involved in aggression as well. But the next critical question that we had was, okay, it might be involved in aggression, but is it actually connected to some of the same mechanisms that I was just talking about, those visual mechanisms? What we found was that, in fact, it was connected to each of those different mechanisms that I showed to you in the female. So all of those mechanisms were involved in, how these aggressive internal states were modulating vision to have an effect on continued behavior. But one critical feature that was different was that actually there was another neuron, P1, which is this other sexually dimorphic cell type, which is actually not found in females. And this particular neuron actually also collaborated with aIPg in each of these different mechanisms. So it really looked to be having this co-regulatory phenotype. So even though there was these similarities, there was also, a difference as well. And, these P1 neurons, I mentioned the fact that, they are actually involved in terms of male courtship behavior, and particularly actually when a male is tracking a female during courtship behavior. And so it was predicted by our collaborators at Rockefeller that they might be involved in terms of modulating visual pathways to have an effect. And, through a collaboration with Vanessa Ruda's lab at Rockefeller, in particular Tom Hiddenmarsh-Stein in her lab, we actually identified that the toggle switch pathway was actually implemented in courtship behavior. And so really what this looks like is actually a more general framework by which these internal states, so not just aggressive internal states, but also arousal, were actually shaping these visual pathways in order to have effect on continued social interactions. And so this is a more generalizable framework for kind of constructing these flexible social interactions. And so we really think that these circuits are recruiting not just these sex variable components, but actually also shared components in order to have this effect on these social behaviors. And so, I've talked a lot about, kind of boosting and continuing aggressive behaviors. And one thing that, I'm particularly interested in is in the downregulation of aggression. So, one critical question to me was, what are the neurons that modulate disengagement, that modulate this retreat phase of aggressive behaviors? And, are they at all involved in terms, or are there any in our circuit of interest? And so, where we got our start with this was actually, when we were searching through the Connectome, what I was thinking about was, these aIPg, these aggressive state neurons, are modulating these output neurons, but what else is modulating these output neurons? And so all of this actually all came out of the Connectome. So what we found was that there was this other neuron, this yellow neuron that I'm showcasing here, which is actually an inhibitory neuron, that actually synapses onto 61% of all of the outputs of aIPg. So it's a quite striking amount. It really appears to be really heavily focused on the aIPg circuit. What it appears to be doing is, because it's inhibitory, it really appears to be shifting down or dampening down this particular circuit. And what we also identified was that, it's not just, that random neurons within this aIPg circuit that it's involved in. Actually, a lot of these are those exact same visual pathways that I mentioned before. That disinhibition, that toggle switch are all here. They're all present. And so it's all going to that. And in addition to looking at the female hemibrain connectome, we also looked within the male, and we found that this was actually consistent across sexes. So it really appeared to be that this particular yellow neuron, which is actually found across sexes, is one that really appears to be actually dampening down this circuit. And so it really looked like a potential disengagement neuron. And so we wanted to test, okay, is this yellow neuron, which is actually called AVLP590, is this neuron, actually involved in terms of aggression? And so in order to do this, we needed a line specific for that particular neuron. And so we again went back to the genetics and did all those crosses. And we got a couple of lines that look like this. So they look very, very strikingly similar to that AVLP590, that yellow neuron. And so what this allows us to now do is we can actually ask, the question, is this particular neuron, does it affect aggressive behavior? And so what I did for this is, again, I boosted up the baseline level of aggression through using those aIPg neurons. And I'm going to show you that here in this video. But what I also am going to do is I'm also going to excite this yellow neuron, because what it actually predicts, what this circuit model predicts, is that when I activate this yellow neuron, which I'm going to do through using optogenetics, so I'm going to do that here. When I activate this neuron, it should serve to actually dampen down aggression if this circuit model is actually what we predict. And so showing you this video here, you can see the flies engaging in aggression, when I'm activating aIPg. And next I'm going to turn on the red light. And so now you see the flies move away from one another. So it really looks like this particular yellow neuron is actually involved in terms of downregulating aggression. And we actually not only just saw this in females, but when I did similar experiments in males, we saw the same effect. And so, what we think this is we think this is a circuit mechanism for downregulating aggression. So, we have these aggressive state neurons that appear to be boosting up and continuing aggression, but there's actually this neuron that is consistent across sexes that is one that really appears to be actually downregulating aggression through modulating potentially the outputs of these aggressive state neurons. And, additionally, we can also use the connectome to see, what other cues these disengagement neurons might be using. So when we look in the connectome, what we find is there's actually other visual pathways that are coming into this disengagement neuron, some of which have been implicated in avoidance behavior. And we also see some auditory pathways coming in. And so this is quite striking, because now we can think about, how multiple cues are being processed in the brain and how that actually can affect these social interactions and potentially this disengagement behavior. And so the way in which I like to think about, this circuit and the circuit model is one in which about a general framework for social decision making. And so, what I believe I've shown you is that, these aggressive state neurons actually appear to be having an effect in promoting aggression and promoting the continuance of aggression through modulating these sensory pathways. And that it appears to be a more consistent feature that is not just found in aggression, but actually appears to be conserved across other states as well, like arousal. Additionally, we also identified this general framework by which, you have this aggressive state neuron synapsing onto the same neurons that this disengagement neuron is also going to. And this appears to be a conserved structure across sexes. And in addition to that, something that I'll be looking into in my future lab is really about the sensory pathways that are coming in. You might have noticed that with those aggressive state neurons, there appear to be differences in the inputs across sexes. And so that's something that's quite interesting, to me, because we actually know that there are differences in terms of the context in which female flies are aggressive versus male flies are aggressive. And so that's something that we're interested in, as well as in terms of those disengagement neurons as well. And so with that, I'd like to thank, my advisor, Jerry Rubin, all of the Rubin Lab, the amazing undergraduates. We have a summer undergraduate program here at Janelia. And so I've been very lucky to have some great undergraduates that I've been working with. And also, this is a team science project. So we have some amazing shared resources at Janelia, as well as with my collaborators in the Branson Autopolic Lab and those at Caltech and Rockefeller. And I also want to mention that, since the male CNS is out, this is actually the preprint for the male CNS. You can dig into, some of the similarities and differences across sexes. And then here's the preprint for our bioarchive from Jerry and from Adrian on these social circuits. And with that, I'll take any questions that you guys have. Thank you so much.
A huge round of applause online. That's a wonderful talk. Thank you so much. It's amazing how you can study such complex behaviors. Before I open up to questions, I want to, of course, thank you, Dr. Katie Schretter, for your wonderful talk again. And also a huge thank you to our audience for joining us today. And I want to take a bit of time to express my gratitude and also highlight some of our ambassadors and partner organizations. So firstly, like a special thanks to the Student Council of International School of Medicine, International University of Kyrgyzstan, and for their team for leading the outreach efforts in Kyrgyzstan, and also Kazakhstan Medical Student Association for the outreach efforts in Kazakhstan. Special shout out to Sohaib, Aisha, Terim, Asad, Ali, Aditya, Sean, Khazer, Muhammad Zishan, Sunwo, and our audience for leading the outreach efforts to their communities from different parts of the world, including Pakistan, Libya, Canada, and the US, and many other places as well. They have hosted in-person and hybrid workshops in different languages, including Arabic and English, and hopefully soon Russian. And also I would like to thank the UCT Cortex Club from South Africa, IYNA Nigeria, IYNA Garbia chapter from Egypt. This is a club, a high school club, U of T Life Sciences Network, Neurovia U of T, and Synapse Club from Toronto, and also the academic office at the Student Council of University of Tripoli in Libya. They have really led the efforts in making the Arabic workshop. So thank you again. And yes, now let's open up for Q&A. We encourage everyone to ask questions, especially encourage high school and undergraduate students to ask questions. I know it might be very scary, but all questions are good questions. And feel free to type in the chat, but also feel free to speak in the mic.
Okay, it looks like we have some. So Abu Bakr asked, how do you simulate those specific neurons on such a micro level?
Yeah, so that's a very, very good question. So the way in which we do this is, we are using genetics to basically precisely mark individual cell types. And then the way in which we can do this is then we can also use genetics to basically insert in specific channels that then those channels, when I shine the red light, then they'll only activate those neurons that have those channels. So then that will cause those neurons to fire. So so it's a very, very beautiful system for that precise marking. So that's a very, very good question.
Hey, Mariana, would you like to ask your question?
Hi, yeah, sorry, 1 second. Okay, so my question was, when you were activating kind of the neurons so they would fire, and like begin those aggressive and also inhibitory behaviors, why specifically does like a red light work, or like why that specific light, I guess?
Yes, that is a very, very good question. So yeah, so the red light works because of the fact that these channels, they have with them a particular, so it's a thing called a channel rhodopsin. So it basically, if that light wavelength of light corresponds to that channel, then that will cause them to fire. So that's the reason why I was using the red light, is because that particular channel rhodopsin is particularly tuned to red light. But actually, when I was inactivating the neurons, I did didn't mention this, but I was using a different channel rhodopsin. So I was using one that was a ligated anion channel that basically what it does is when I shine green light, then it inactivates the neuron. And so the green light is, that channel rhodopsin is tuned to the green light, and then the other one is tuned to the red light. And so, we can kind of use some of the chemistry to be able to work to our advantage for that. And that's the reasoning for that. But yes, a very, very good question.
Okay, thank you.
Okay, so Muhammad had a question. Why Drosophila is a suitable model for this kind of studies?
Yeah. So for Drosophila, like the reason why I think Drosophila is particularly suitable for these types of studies is, we have a lot of very beautiful tools. So the tools I was mentioning in genetics, and we also, because we have these tools, and also the flies exhibit, very complex aggressive behaviors. So they exhibit these aggressive behaviors. We have those genetic tools, and also now we have the full map of the fly brain. There have been maps that have been generated for other organisms before, I should mention. So, C. elegans, so worms actually have a map as well, but they don't exhibit complex aggressive behaviors, to my knowledge. So we have, now the capability to go in with this particular organism to be able to ask those key questions about, how does the environment shape these particular, social circuits in the brain? So how do they shape all the neurons in the brain in order to have this effect on social behavior? And so that's the reason why I think is because of all the tools that we have and because of the connectome. So we can now be able to generate these beautiful hypotheses, and we can generate these hypotheses and think about them in many different ways.
Wonderful. So Salman, would you like to ask your question?
Yeah, thank you so much for the opportunity. I would like to ask, how is the decision between aggression and disengagement made?
Yeah, no, great question. Yeah. The decision between, we don't really, know exactly, this actually gets into a really fundamental question that we don't know a lot about. So we don't know a lot about how exactly these are being, the switch is being managed between, the going into an aggressive state versus, going versus disengaging. It's something that I'm particularly interested in is, how exactly these sensory pathways might be modulating that switch. And that's something that, across many different species, people are starting to look into. But in the fruit fly, we now have the whole brain connectome. So we can be able to look at particular cells and really mark those particular cells and really look at how exactly they are regulating this. And we can also image from these aggressive state neurons while the flies are, tracking different moving objects, as well as this disengaged neuron to be able to see, when the activity peaks and when it kind of dips down. And so we can do those experiments now, but it's a very critical question that we actually don't know exactly the answer to. So that's very good question.
Wonderful. So we have a question from Naila and Fatima, and it's similar, so I'll just group them together. This research topic is completely new to me and leads me to ask, how might the insights from this work help us understand similar behaviors or neural mechanisms in humans, or would it be completely different?
Yeah. So there are some, so when we think about flies offer up a unique opportunity. And this is a very, very good question. It offers up a very unique opportunity for us to think about it on a simplified level, because the fruit fly brain, is very compact. And so we can think about, how exactly these are general principles that then can scale up. So we do in fact find that there are similar circuit principles found in similar, kind of connectivity types of connections that are found in mammalian species. Work that has been done in mice has identified some beautiful work in terms of, how exactly sensory information might be interacting with aggressive internal states. But we can't get at, kind of the very, very, the individual cells in the mouse brain. We can look across many different regions, but it's really really, really hard to do lots and lots of genetic crosses in mice. And so, for us, what this can give us is it can give us a general framework for how this might work in mammals to then be able to scale this up. And so that's something that I'm thinking about for, long term, is thinking about, how exactly might this be, might this be an example or, or regulated in, mammalian systems that might be more complex. So while there could be some fundamental principles that are the same, there probably are some unique features that are different across species. And so that's something that, I'm particularly interested in is, how exactly, are these differences? Because we know that Drosophila in and of themselves, across different Drosophila species, they have different aggressive behaviors. And so you can kind of do those across species comparison and see what are the fundamental principles and then what might be more species specific.
Wonderful. I'm seeing lots of questions. Amazing. I promise I'm not ignoring people. I'm going to try and give priority to the people who didn't ask. So just keep that in mind. So you may go ahead and ask your question.
Hello, thank you so much for this wonderful talk. So I have a question about the internal state, the aggression internal state that you have suggested about that the aIPg neuron is regulated. So I have a question. So you suggest that these neurons are responsible for regulate the internal state, the aggression internal state of the flight. So I have a question. So when you look at the electrophysiological data of maybe downstream neurons such as like LC10C neuron, are there any property that suggests that when you stimulate the activity of the aIP neuron and then you stop those activation, are there any effect but you don't let the fly engage in the aggressive behavior? What are the new electrophysiological data of the downstream neuron? Are they exhibit like increased activity when you like prevent them from engaging with in aggressive behavior that reflect the internal aggressive state being still like accumulated and not satisfied by engaging in those aggressive behavior?
That's a great question. And actually we don't know the answer to that question. So we haven't yet recorded from those downstream neurons. There has been some work that actually recently has been published, I think maybe only like 2 days ago, by another group and at Harvard, where they did, Rachel Wilson's group at Harvard, where they did record from downstream neurons. These were neurons in the AOTU, which is the anterior optic tubercle, which some of you guys might be searching in. Those neurons actually, they do see that they are modulated by aIPg neurons, but we don't know very much about, this during behavior function. So, and we don't know very much about other neurons outstream. So like all of those output neurons that I was talking about, the LC10C, actually LC10C, we don't have a specific line just yet for that particular neuron. And so, some of these we don't have, we weren't able to, to get genetic access to those particular neurons. But the ones that we do have genetic access, that is something that I'm looking into, that I'm interested in looking into, is how exactly are they modulated? How are they modulated during aggression? So this is a very, very much of topic, and exactly in line with what I was thinking, is trying to go down that trajectory. So that's a very good question.
Excellent. Let's go with Terim next.
Yeah. Hello. Okay, so what I'm meaning to ask is like we use red lights, like you use red lights, different wavelengths. So like any other experiments have been done using various wavelengths and seeing how they are like affecting, and are they affecting other neurons? Like, does red light only activating aIPg or P1? And another, like the same neuron, is it involved only in one function, in just aggression, or are there potential for any other social behaviors, or no? Like, or another thing, that like only PG1, aIPg or P1 are involved in aggression, or no, there are other neurons that have potential for similar functions, or like this?
Yeah, no, these are wonderful questions. So answering your last question first. So in terms of these aIPg and P1 neurons, they very much might be involved in other behaviors. We don't actually, I had done some courtship experiments with the aIPg neurons. The data was very variable, so I didn't, it would need to be looked into a lot more in depth. But we don't know very much about, in the wild, when is aIPg on and when is aIPg off. It's been predicted for P1 that it's definitely on during courtship behavior and on during aggression. But exactly how I was talking about these different types, we don't know very much about how each of these different types might be contributing to different parts of the behavior or might be contributing to other behaviors. And so that's something that I think is of huge importance and is very, very interesting to look at. And then in terms of the first question about the red light, so looking at other wavelengths, like the wavelength that I'm looking at is shifted very, very far red. So it's roughly around about 655 nanometers. And so that particular wavelength is particularly suited for that channel that we're looking at. And so that's the reason why we pick that particular wavelength. Now, if you look at another wavelength, say if I shine green light, that's slightly within the range of those for those particular neurons, you would see an activity, but it would be lower. And so we don't use, that's the reason why I don't use green and red right now, is because of the fact that, it's hard to kind of, like there can be some overlap between those particular wavelengths that you can see. The specificity, you shouldn't see any other neurons being activated since we are expressing it only within those aIPg or those P1 neurons. And so we don't see activity elsewhere. And so actually when I showed you the image of the aIPg neurons, it was only expressing in those particular neurons of interest, which suggested to us that it was specific. So there shouldn't be activation of other neurons. However, you're shining a red light, and so obviously that's going to create some startle effect of the flies. So you might, if you shine too much red light, then you can see some type of startle effect. So because of the fact that there is a visual stimulus there. And so, we are very, very careful about making sure that the red light is lower for that reason. So that's a very, very good question about, the technicality, because sometimes the technicality can be harder of the assay. So thank you.
Great.
Wonderful.
Let's go, Mariana. Feel free to go again.
Hi. So when you were talking about like aggression and near the beginning, you mentioned aggression, but you also mentioned aggressive internal state, and I was just wondering what the difference between that was, because I kind of understood that aggression you meant as like outward aggression, but then for aggressive internal state, I wasn't really sure like how you can kind of measure that or what that kind of means.
Yeah, no, that's a very, very good question. So you're correct that the aggressive behaviors, I was meaning the outward aggressive behaviors, so the things like the headbutting and the fencing. The internal state, it gets complicated because of the fact that I'm using the same word for it. But really what it means is that it's a hidden, basically a hidden state. So it's something that the brain kind of has that is like usually more distributed across the brain. But these hidden states are things like hunger, arousal, and aggression is thought to be also another internal state. So kind of you can think about this as kind of, if you like anger, so kind of like you build up the anger. But because of the fact that, it's flies, I can't ask them whether they feel angry. That's the reason why we talk about it in the context of like aggressive internal state, is the reason that we talk about it in those terms rather than using the term anger, or kind of the more like, emoting types of terminology, if that makes sense. But that's a very, very good question on it, since it is a difficult concept. If that answers the question, hopefully that answers.
Yeah, it does. Thank you so much.
No worries.
Great. Before we go to Salman, I'll just ask two questions in the chat because it's been a while. Abu Baker asked, how do you navigate that specific aggression neuron? But I want to rephrase a little bit, which is, how do you actually do those specific cell lines or like genetic lines? Like, what does it mean to have a genetic line? And I think that question also answers why Drosophila might be a very good animal model. So if you could address that, that would be great.
Yeah, of course. So the genetic lines, like what we're doing is we're basically we're taking so the way in which this was first constructed is that, the history of this is that what you could do is basically for flies, they have really, really great life cycles in terms of the fact that, very short life cycles. So what you can do is you can actually then put in specific inserts into the fly. So you can genetically manipulate them. And so what we're doing is we're basically putting in inserts where what we can do is we can basically then have it being that we're essentially only labeling specific cell types, if that makes sense. So we're essentially just tagging specific cells. And the way in which we now do those tags is we actually do them through using kind of some other basically kind of like toolkits, if you will, that were actually first developed in yeast, which is like a combinatorial method. So we're basically taking, one particular yeast product, which basically what we can do is we can break that yeast product into parts. So one part is called it's an 80, which is basically it's a particular adaptive domain, and then we also have a DVD, which is basically so it's just two domains of the same component of it. And so what we can do is we can express them in two different places within the fly genome. And then what we can do is we can use that particular labeling and intersect those two so that now when they come back together, they're now only then expressing in one, if that makes sense. So really what we're doing is we're basically using some very elegant combinatorial methods. If you think about this more in the context of kind of mathematics, what you're doing is that you're basically taking two mathematical terms and basically looking at where exactly they overlay, so where they intersect. So kind of doing a Venn diagram of this. And that's the way in which we go about doing the genetic structure of it. And so then what we can do is because we can basically take, different of these particular, of these two particular components of these two particular, basically this AD and this DVD, we can take them and we can use them in different ways. So we can link them to different factors and then express them in different areas of the different neurons through using things called transcription factors. And so then when we do that, we can then intersect them in different combinations. And so we do this kind of combinatorial method of doing those genetics. Does that answer the question of it? It's a more complicated answer to that question, but that's the way in which we go about doing it, is kind of like an additive way of looking at who exactly might be a good marker for those particular neurons. And so the reason why this is, such a added benefit with the fly is because of the fact, in mice, they're, in terms of, their reproductive cycle and also in terms of their, lifespan, their lifespan is much, much longer. Their reproductive cycle is longer. So, it does take more time, and also, they generate less in terms of their offspring. And so flies generate a lot of offspring. We can do this on a very, very high scale so that we can basically have a lot of different, we can do all these things that I was talking about with crosses. So we can do all these different combinations on a very large scale. And that then allows us to be able to then go through many different cycles in order to generate one particular line that is a genetic line that only labels that particular cell type of interest. So, just for scale, it took roughly around about 50 crosses. So 50 of these different combinations of me, crossing different lines and then imaging those particular flies in order to actually generate two lines that labeled those IB112 neurons. So it can take a lot of going through all this, and flies also are a lot cheaper. So, mice are a lot, I used to work in a mouse lab, and mice are a lot more expensive than flies are for doing this. And in terms of genetics, we can also do a lot more combinations. So, the things that I showed you where I was doing both optogenetics and thermogenetics, so I was using both light and temperature, that actually takes 5 different things that I'm inserting into the fly genome in order to make sure that those flies have the correct genotype for me to do those experiments. So that's a lot for those flies. And so that is more easily done in the fly, if that makes sense. If that's kind of a better
Yes.
way.
Hopefully, Abu Bakr, that answered your question in detail. And maybe Salman, go ahead.
Yeah, thank you once again for the opportunity. I would like to clear one doubt that why the male flies, they are only showing the aggressive behavior. Why only the male flies?
So the female flies show aggressive behavior as well. So they show different aggressive behavior. So it's just a slight difference in what exactly they do. So the female flies will do, and the primary bulk of what I showed in the videos were all female flies. There was only one video of male flies. So the female flies show the aggressive behavior, but they just show it differently. So this is a really good question because of the fact that actually, when I showed it at the beginning, the female flies were doing the headbutting interaction, and that's primarily found in the females. And the males were doing this lunging behavior. Now, the females don't really tend to exhibit, at least in this species, don't tend to exhibit the lunging behavior. We don't know why. So that's a very, very good question. We don't know what the basis is for that, but we hope that, through having these 2 connectomes, we might be able to see maybe there are certain neurons that might actually be producing these differences across sexes. But we do know that, for those neurons that I was looking at, these aggressive state, these aIPg neurons, we do find that, while they are different in morphology across sexes, they do appear to actually be having an effect on aggressive behavior in both sexes. And so that is quite nice for us. So we don't know about maybe there are certain neurons that might be different, or maybe there might be, many different neurons that might be contributing to this. And we don't actually know about that. So it's a very, very good question.
Thank you so much. Thank you.
Great. One question in the chat, Fatima asked, do you see potential applications of your findings in psychiatric or behavioral neuroscience research? I think you kind of answered this, but maybe if you have another.
Yeah, no, it's a great question. I mean, I think that, it's something that I'm particularly interested in is, I came from a lab in grad school that was very much dedicated towards thinking about this in terms of therapeutics. The way that I like to think about it is, if we can have some fundamental principles, if we can figure out this circuit, and then maybe also look at, some mutations that we know are involved in different neuropsychiatric disorders. So there has been some really, really beautiful work in humans to look through and look at the genome to see, whether there are certain markers that might fall out. They've seen that, it's not as simple as one particular marker that's falling out for a particular genetic disorder, for many different particular genetic disorders. And so we might actually have to do multiple different mutations. And that's something that we can do in the fly. We can actually do, different point mutations in order to see, whether those point mutations in specific neurons might contribute to different social outputs. And so that is something that I think, could have, broader impacts for kind of understanding how exactly these neuropsychological disorders, how they come about, as well as, how just differences across individuals and, differences in terms of context really have an effect across different individuals. So, we know that life history can have an effect on social interactions, and that's something that, has been heavily studied in psychology. And, we can kind of try to get at that as well on a circuit level using the fruit fly and see if there's any generalizable principles. So I'd be very, very interested to work with people who are interested in that type of a focus. So it's a great question.
Okay, wonderful. Vipul, would you like to ask your question?
Thank you again for the talk. So I have a question about do you have any data or hypothesis about what is the neural circuitry that responsible for the observation that after the fly engages in aggressive behavior, it will reduce their aggressive internal state? Like if not, they will continue to be like stuck in the aggressive internal state. So do you have any like um hypothesis or circuitry that may responsible for this reduction of internal state after it engages in the aggressive behavior?
Yeah. So like kind of like what the internal state is that they might switch to, which is I mean, I think that that's a very, very interesting question, and a very good question. We don't know a lot about, like in terms of the behavior of what happens afterwards. It seems like they just, move away from one another when I'm activating the disengagement neuron. I don't know if that is also the context. You might have noticed that, in my in the particular, behavioral arenas I'm using, it's very, very bland. There's really not that much there. There's really only the other flies. There's not, really other cues. There's not other species. There's really nothing there. So it could be that, maybe I'm not able to actually, in my current setup, I'm maybe not able to, see what else comes out when they disengage. And so that's, I think, a really interesting thing that I've been thinking a lot about is, like when they're in the wild, there's lots of other things that are around. So there's lots of things that they could attend to otherwise, and lots of other, features that might be important. They can also fly away in the wild. In my setups, they cannot. So that's another factor that is something that I think would be really interesting to look at. And we did do at the beginning of my postdoc, we did a couple of studies with someone working in escape behavior. We didn't find anything specific, but that doesn't mean that, I mean, we only did cursory studies, so we really could see that there could be something involved there if we looked harder, and so that's something that I would be interested in. Since there are others who have looked at, courtship versus, versus kind of this escape behavior and seen some really interesting phenotypes there. And so, I think that is a really interesting field of study. If that answers the question, I mean, the hypothesis is that there probably is some other states that come up. So this aggressive internal state doesn't, is probably not in isolation, is my personal hypothesis. It's probably that, there are different gradations of, hunger, of, all of the arousal of all these other things that are going on, that are all happening at the same time. But it's just, to me, it seems like, one of them might pop up and be more important than the other one. There's been lots of proposals. I mean, Tinenberg and Lorenz put forward there are these kind of big names in neuroscience that put forward these very, very beautiful hypotheses for how this functions. And I'm not sure, in terms of the fly, I think that's actually a field that's really ripe for study, is like, how exactly is this, are these different states, how are they competing? And there's a couple of groups, one at the University of Birmingham that's looking at this. I think that is a very exciting field of study. So yeah.
Wonderful. Okay, sorry, because of time, we need to be a bit mindful to end a bit early. So I only have time to take up 2 questions. So because Nafan and Mariana, you've been waiting patiently, I'll let you do that. So first, I will address Nafan. I hope I'm saying your name right. Your question. So the question is, does the number of wavelengths affect the flies of male and female? How big is the difference?
So in terms of the wavelengths that we're using, that's a very good question. So in terms of the wavelengths that we're using, we're using the same wavelengths of light for this. But actually, it's a very striking question because I didn't mention it. But when I am activating this disengaged neuron, I actually find that different like amounts of that wavelength are actually important. And so this, it could just be that it's, simply just, something that has to do actually that's, not having to do with this particular neuron of interest. It actually has more to do, maybe with the opsin that I mentioned before. But if we record from this neuron, there might be some differences that we could find. And that's something that I'm really curious about, is about, whether or not there are differences in this disengaged neuron that actually we are seeing in the behavior that maybe actually aren't a feature just of the technicalities of the experiment. And so that's something that I'm really interested in. So yeah, we do see some slight differences, not in the wavelength, but actually in the amount of the light that we're delivering. And that is something that I think, and actually in the timing of the behavior too. And so that's something that I think is really, really, really exciting. So it's a very, very good question.
Hey, thank you so much. So Mariana, you may go ahead and ask your question.
Hi again. So my question was, in the experiment that you ran when you separated the two flies, but still activated kind of the neurons responsible for internal aggressive states, you timed it for 10 minutes and then released them. Is there sort of like limit to how long it lasts?
Yeah.
Or is and what's the minimum, I guess?
Yeah, no, that's a very, very good question as well. You guys are asking phenomenal questions. So I didn't mention it, but there is a limit. So actually, if we extend this out to 30 minutes, you don't see a significant effect. So it really looks like this is an internal state that lasts, but it doesn't last forever. It lasts for tens of minutes, but it doesn't necessarily last beyond 30 minutes. And then the shortest amount of time that we did was 1 minute. And we did see aggression, but that was almost an immediate kind of switch that we were looking for there. And so we did gradations from 1 minute, 5 minutes, and 10 minutes, and then 30 minutes. The 10 minutes was the least variable one that we found. So there was a higher amount of variability at 5 minutes, which is interesting. It could just be the flies that we were using. It could have been something having to do that's not having to do with the internal state. But it could also be something having to do with the internal state. It could be that the internal state gets more fixed and then becomes more fixed at 10 minutes. And that could be something that could be there. So you do see, you don't see like a continuation for forever, but this is actually, what we don't know is we don't know across sexes, whether there is, number one, we don't know if aIPg in males, I said it, but I said that, it was aggression, but we don't know if it's actually aggressive internal state in males. There's other neurons that are known to be involved in terms of these internal states in males. And so we don't yet know about, whether aIPg is kind of doing this effect, the exact same effect across sexes in terms of those states. So that's an extremely good question as well. So yeah, very good questions. You guys are doing phenomenal questions.
Perfect. Thank you so much.
Hey. Wonderful. Yeah, thank you so much for asking so many incredible questions. I learned a lot and I'm sure everyone else did. And yeah, this concludes this wonderful talk. Thank you so much, Dr. Schretter, for sharing all your wonderful work with us. We learned so much, and obviously thank you for everyone for being a part of this journey. All of you have made this so exciting, and I think this was really, really cool. Yeah, and if you have any last words, Katie, we would love to hear them.
This was phenomenal, and thank you guys for the amazing questions and and for attending. It's a phenomenal experience to be able to talk with so many of you guys, and wonderful to have such great questions. I mean, and such great interest, and also, I'm really looking forward to all of the amazing work that all of you guys will do with the Connectome. So, it's a great program, and I'm very, very excited to see, all the results. So wonderful work and wonderful job on all of this. Thank you guys so much.
Thank you.
Bye-bye, everyone. Thank you so much.
Bye, everyone. Take care.
Bye.
Clematis Research Empowerment Hub. Empowering research for everyone.